Bismillahirrahmanirrahim. Now we are going to start the reproductive section of our the reproductive unit of our sessions. So in this session, initially in the PDF which I am using of the first aid, they have mentioned some uh, very horrible sort of embryology with all the genes and stuff like that. It is not mentioned in the first aid original version, but it is very important and it is very interesting as well. So we will start with that, and I will like to add a video in the I button. This is very wonderful video, and that explains this concept very well. I will try to explain it as much as I can, but for the further details and for a better explanation, please go to this video link. I'm like that will be visible in the I button. So I will also put the link in the description. So let's get straight into it. The limb development or limb patterning. Limb develop along the three parts, that is proximal to distal. That is, suppose we are standing with our arms extended, so if we are moving from the humerus towards the wrist, that is, we are moving proximal to the distal direction. Like suppose that if there is a person over here who is standing like this with his limbs extended, so if I'm extending in this direction from the humerus towards the wrist, I'm moving from the proximal to the distal direction, firstly. Secondly, the dorsal ventral axis. The dorsal is extensor versus ventral is flexor. Suppose you look at your hands, so the palmar surface of your hand is the ventral surface, while the opposite surface is the dorsal surface, the back side of your hand. Same goes for your feet, and then it applies to every part of your body, with the ventral being the it applies to all limbs of your body, with the ventral being the flexors and dorsals being the extensor surfaces. Then the anterior posterior axis, that is a bit confusing. So anterior towards the head, which is radius and thumb. Suppose that a person is having his arm extended like this is the thumb, and here are the fingers, 1 2 3 4, I guess we're done, and then these are the arms. So if the person has extended limb, so this particular direction, the thumb, and with the thumb we have the radius going inside, so thumb and radius are anteriorly placed, while the ulna and the finger, or the pinky finger to be specific, that is placed posteriorly. So that is something that is confusing, because it is not according to a very conventional orientation concepts. So remember this one is very important. Then proximal to distal development. Now coming to this diagram is basically representing the limb development. So we know that, suppose that this is the fetus, and fetus looks something like this, so there is emergence of limb buds. Like for the upper limb there are two limb buds on each side, and for the lower limb there are two limb buds on each side. This is a limb bud that is represented in this picture. The AER, or the purple zone, that is apical ectodermal ridge, because we know that whenever there is development, I will try to make it with three colors. So if you have listened to the Dr. Najib lectures, it will be clear for you. But suppose that let me draw it here. Suppose that here we have a cut section of the embryo which looks something like this, and then it is like this, and then we make it something on those lines like this, and here is the yolk sac, and it comes like this. So suppose that the upper surface, or the upper layer, is the ectoderm, the inner cells, they are the endoderm, and the cells which are I'm going to draw in between both of them with say purple color. So these color, this gooey material between these two layers is the mesoderm, right? So I have drawn this picture just to make you oriented to the three layers of the embryo. So embryo contains three basic germ layers, which are ectoderm and mesoderm and endoderm. So the purple one, that is basically the ectodermal layer. Then comes the ZPA, and that is zone of basically not the zone of polarizing activity. The mesoderm is something else, that is called as apical, that is something else. I guess that is polarizing zone or something like that. Sonic hedgehog for the patterning or something. Okay, so zone of polarizing activity, this is this one, and then the progress zone, I was talking about this one. So let's look this picture. So the ectoderm is basically forming the apical ectodermal ridge. Below that we have the mesoderm, that will be forming the progress zone, and then this particular zone is also basically coming from the mesoderm, that is called a zone of polarizing activity. So just remember these three zones. So the outermost, so the AER, that is the ectodermal covering, and under that we have mesenchyme, and mesenchyme is consistent of the two further zones. One is the progression zone or the progress zone, while the second one is a zone of polarizing activity. Now the apical ectodermal ridge, like this one, it is responsible for the linear growth of the limb, and if it is removed, the limb growth is halted or inhibited. So proximal to distal development, which means suppose that we are talking about this development moving from humerus towards the wrist, or the limb bud that has emerged from the embryo, now it is moving from the proximal to the distal end. So how is that taking place? It depends on the apical ectodermal ridge, because I talked about it that for the lengthening of the lengthening of limb, we are dependent on the apical ectodermal ridge, or AER. Ectoderm overlying the mesoderm. So okay, it is ectoderm that is overlying the mesoderm. Area of limb bud formation. Removal will lead to limb stop growing. So if we are going to remove the AER, the limb won't grow. Influences underlying the mesodermal growth. Key transcription factor is fibroblast growth factor. So in the AER, the key transcription factor is the fibroblast growth factor, which is symbolized like over here. Where is that symbolized? It was somewhere here. Okay, it is drawn within the picture. If you can see, FGF is written over here. So the main transcription factor during the growth from proximal to distal direction is the FGF, and the FGF8 is such a potent transcription factor that suppose we talk that if we are going to remove the AER, there will be no growth, but if FGF8 is present in ample amounts, it is enough to compensate for that growth. That is just a side note. So if you want to forget it, please forget it. The thing that you must remember it is that for the proximal to distal growth, the most important transcription factor stands out to be fibroblast growth factor. Then the dorsal to ventral development, which means that the palmar surface and the dorsal surface of the hand. So the development in this axis is dependent on the WNT7 gene, and that is the key for the dorsal development. Okay, and then anterior posterior development, it depends on the zone of polarizing activity. This one, like the ZPA, the zone of polarizing activity. Zone of polarizing activity will determine that which limb is going to grow from the bud which has emerged or which has sprouted out of the embryo. Suppose we have two buds for the upper limb and two buds for the lower limb. So it is a zone of polarizing activity that will determine that this bud will form upper limb and the other bud is going to form the lower limb. If we replace the buds with each other, if we replace the zone of polarizing activity of ZPA between the upper limb and the lower limb, so lower limb will start developing in the place of upper limb due to this zone of polarizing activity. So they have mentioned that the anterior posterior development, it depends on the zone of polarizing activity, and major signaling molecule is sonic hedgehog protein. So the thing that the takeaway point from this page is, very first thing is that you must remember that what are the axes of growth. The proximal to distal, that is from the humerus towards the wrist. Dorsal ventral, that is flexor and extensor differentiation, and the third one is anterior posterior axis, that is the thumb is your anterior thing and the pinky finger is your posterior axis. So now the second thing that you must remember from this page is which are the transcription factors or the areas which are responsible for growth in these particular directions or these particular axes. Proximal to distal axis, the transcription factor is fibroblast growth factor, and it is dependent on the apical ectodermal ridge. For the dorsal ventral development, WNT7 gene will play a role, and that plays a role for the dorsal development, dorsal to ventral development. Okay. And for the anterior to posterior development, we have zone of polarizing activity, and major signaling molecule is the sonic hedgehog gene. Then limb development, proximal to distal dependent area is the apical ectodermal ridge, and fibroblast growth factor gene will be there. Let me remove this one. Okay, so why did you, okay, we got it. So okay, so for the dorsal to ventral, it is also dependent on the apical ectodermal ridge, and WNT7 gene is playing a role in that. And anterior to posterior axis limb development, zone of polarizing activity is the dependent area, and sonic hedgehog gene and the homeobox gene, also called as the craniocaudal gene, will play its role. Then we have important gene for the embryogenesis, sonic hedgehog gene, embryonic signaling protein. So what are the important genes? Now we will look at them one by one. Sonic hedgehog gene is embryonic signaling protein. It is a very important gene in a lot of structures, and specifically for the limb development and the notochord, uh, in the neurulation process, from which the notochord will give the signals to the upper lying ectoderm to conform that into neural ectoderm and neural plate and then neural folds and stuff like that is formed. So the whole concept of neurulation, in the whole process of neurulation, sonic hedgehog has a very important role, and same goes for the limb development. It is involved in the patterning along with the anterior-posterior axis. So the anterior-posterior axis, which is this one, okay. So for the anterior-posterior axis, which is dependent on the zone of polarizing activity, the sonic hedgehog is going to play its important role. Limb development, produced at the base of limb in the zone of polarizing activity, as you can see in this picture. This is ZPA, and sonic hedgehog is protein produced over there. It is a morphogen. And for the CNS development, formation of forebrain, that is prosencephalon, signaling separates the right and left brain. It establishes a midline. Mutation can cause holoprosencephaly, single lobe brain, that is very important. I guess it is with the fetal alcohol syndrome that we find a holoprosencephaly picture, and that is happening because of the mutation or some interruption with the sonic hedgehog gene pathway. Okay, then we have WNT7 gene. It is produced at the apical ectodermal ridge, thickened ectoderm at the distal end of each development ridge. So thickened ectoderm at the distal. So what is apical ectodermal ridge? As you can see the purple one in the picture, that is going to produce WNT7, and WNT7 is produced from the apical ectodermal ridge, which is actually a thickened ectoderm at the distal end of each developing limb, necessary for the proper organization along the dorsal ventral axis. Fibroblast growth factor gene, produced at the apical ectodermal ridge, stimulates the mitosis of underlying mesoderm, providing for lengthening of the limbs. So the fibroblast growth factor gene is produced at the apical ectodermal ridge, and that is responsible for the lengthening of the ribs. WNT7 is also produced over there, but it is responsible for the dorsal ventral axis. FGF causes lengthening of the rib. So look at the fetus growing fingers. So what is FGF? Fetus growing fingers, which means that the growth of the fingers, or the lengthening of the fingers or limbs, is dependent on the fibroblast growth factor. Then we have homeobox or the Hox genes. It is involved in the segmental organization of the embryo in the craniocaudal direction. Homeosis, transformation of one structure into another. Homeotic genes lead to formation of the body segment. So you know that body is divided into a lot of segments, and those segments, I guess, I'm not sure about them. So these are the homeobox genes. So think of it like, so when we did the neuro section, we talked about dermatomal distributions. So the body is divided into a lot of dermatomes from head to toe. So in the same way, the embryo is divided into a lot of segments, and each segment is a homeobox gene. Each segment is dictated by, the growth of each segment is dictated by the homeobox genes that will lead to the formation of a particular segment of the body. Okay, mutation, okay, that involves the segmental organization of the embryo in a craniocaudal direction. Mutation, appendages will be in the wrong location. For example, fruit flies, legs grow from head instead of antenna. Polydactyly, extra fingers and toes, and syndactyly, fused fingers and toes. So if anybody is asking you about polydactyly and syndactyly and the gene involvement for that, you must go for the homeobox or the Hox genes. Code for transcription factors. Okay, then we have this one. So here you can see that sonic zone of polarizing activity of ZPA, that is going to produce sonic hedgehog, that leads to the CNS development and holoprosencephaly. And head box of the body, that is responsible for the segmentation. This head box is the homeobox, this one. So that is responsible for the segmentation of the body, and both of them are responsible for the growth in the anterior-posterior direction. Anterior-posterior direction. Anterior-posterior direction? I don't think so. We're talking about fibroblast growth factor. Fibroblast growth factor is responsible for growth in the where is it? It is in the proximal to distal direction. So it is basically the AP is for the apical ectodermal ridge. So AP is the apical ectodermal ridge. WNT7 is for the ventral to dorsal growth. No, I was right. I'm sorry, I was zoned out. Sonic hedgehog and the head box or the homeobox are for the anterior-posterior growth. WNT7 is for the ventral to dorsal growth. W in the is for the ventral to dorsal, and sonic hedgehog and head box is for the anteroposterior, while the fibroblast growth factor, fetus growing fingers, that is for the proximal to distal. So if you want to remember only one thing until this time, in everything which we have studied till now, so remember this table, that sonic hedgehog and head box or homie box, they are for the anteroposterior axis growth of the body. Sonic hedgehog is present in the zone of polarizing activity. That also helps in the CNS development, and if that is damaged, there will be hollow prosencephalon. And head box is the box of the body and causes segmentation. WNT for the ventral dorsal growth, and FGF is for the proximal to distal growth. Then we have this picture over here. So I just want to highlight the role of BMP. So if you can see this, like if I could zoom it in. So here in this picture, you can see that the zone of polarizing activity is producing sonic hedgehog, okay, and then it will be responsible for the anteroposterior axis growth. That is not our concern right now. So it is producing the GRE protein, or the GRE, it is gram gene basically, that normally inhibits the bone morphogenic protein or BMP, because when the limbs are growing, so in that particular position we want the FGF to be in excess for the limbs to grow to the ample or optimal length. But once the FGF is enough and the limbs have grown to their ample length, then FGF needs to be stopped. So that FGF is stopped by giving FGF will give allostatic signaling to the sonic hedgehog, and sonic hedgehog will inhibit the gram protein. If the gram protein is inhibited, resultantly the bone morphogenic protein will come out of suppression. That you can see that GRE is suppressing the bone morphogenic protein, and when the bone morphogenic protein can suppress this one, the very concept that I wanted to highlight, and the reason why I came to this picture is that bone morphogenic protein is responsible for the inhibition of the bone growth, and it is responsible for inhibition of the lengthening of the limbs, and it does so by inhibiting the fibroblast growth factor. So let's come back to our topic. We did this, right? Okay, now let's move on to the embryogenesis. So chromosomes versus chromatid. So a chromosome is made up of two chromatids which are joined by the centromere. Okay, this is a chromosome, and the two chromatids will be joined together to form a chromosome which are joined by the centromere. The chromatids separate from each other during the mitosis to form two new chromosomes. The DNA making up chromosome is dispersed as chromatin. So these are basically this paragraph is just symbolizing the terms which can be confusing at times. The first thing is chromosomes. So these four strands, like two upwards or two downwards, or the whole structure like this one is a chromosome which is consistent of two chromatids. Like this one is one chromatid and this is another chromatid. They are fused together to form a chromosome, and they are joined together at this particular point, and that is called a centromere. The chromatids separate from each other during the mitosis to form two new chromosomes. So when they will split at this particular point, and two new chromosomes will be formed, right? Now there are some particular concepts about this one which we will study later on when we will do the spermatogenesis and the oogenesis, and I will explain them over there. But the DNA making up the chromosome is dispersed as chromatin. So when the cell is not dividing, so within the nucleus we have very fine thread-like structure that is not oriented or arranged in the form of a chromosome, and that particular thing is called as a chromatin. So remember that. Then talking about fertilization, how does fertilization take place? Haploid mature spermatozoon 1N 1C fuses with the haploid ovum 1N 1C, forming 2N 2C. So when do we call it 1N 1C? So in human beings there are 23 pair of chromosomes in each cell, right? So when we are diploid cells, like we are diploid beings, and every cell of the human body is diploid. So this particular conformation is called as 2N. That suppose the number of chromosomes, N is the number of chromosomes, and we have two copies for that, so we call our cells 2N, right? So normal somatic cells are 2N, but whenever we are going to form the gametes, so the gametes or the germ cells, they are haploid cells. because normal somatic cells are not going to participate in the fertilization, and there is no fusion going to take place. But in the case of gametes, the sperm is going to fuse with the egg. So both of these cells are supposed or need to be haploid, so that the cell which will form after the fusion of both of them will be diploid, right? So this is the concept about N. So the N is about these cells being diploid, containing the it is about these cells being diploid and containing double the number of chromosomes. Okay, so haploid ovum 2N 2C. So what does it mean when we call 2C? Suppose that I'm talking about chromosome number 2. We have 23 chromosomes. I'm talking about an autosome, yep, somatic chromosome or other chromosome number 2. Suppose I'm talking about that. So in every cell of my body, there will be one paternal chromosome and one maternal copy of that particular chromosome, right? In this particular thing, in which one chromatid is the representative of a chromosome normally in the biology books look like this, but it is the duplicated form of a chromosome. It is not actual chromosome. Normally the chromosome which is present in an in the non-dividing somatic cell of our body, if the chromatin has condensed itself into a chromosome, so, or you may say that right after replication and before the chromosome has decondensed into chromatin, so the chromosomes are like this, like a single chromatid is representing a chromosome. But when this particular cell has to divide, this chromosome when this has to divide, so this particular chromatid will start duplicating, and if it is duplicating, so these one chromatid will become two chromatids, and this particular chromosome will also become two chromatids. So now we have total 4 chromatids for the chromosome number 2. I will repeat it. Initially, okay, so if it is confusing, so I will make it chromosome number 1, because 2 is like a lot over here. So suppose I am talking about chromosome number 1, right? And in my suppose I am taking a skin cell of my body. So in the skin cell of my body, that is a somatic cell, I have 2 copies of chromosome number 1, that is, one is from my father, paternal chromosome, and one is from my mother, that is maternal chromosome. Clear? First thing. Secondly, this is what a chromosome looks like in an adult cell, like it is a single chromatid chromosome, right? Now how many chromatids do I have for chromosome number 1? That is 2 chromatids, so I call it 2N 2C. I'm sorry, so it is called as 2C. I'm sorry, so it is 2C. Like I have 2 chromatids for any particular chromosomes, like for the number 1 chromosome I have 2 chromatids. Now when this chromosome starts dividing, so the chromatids will duplicate, like it will become something like this, and same goes here. Now count the number of chromatids which I have for this first chromosome, that is 1 2 3 4. Now 4 chromatids are present for one chromosome, for the chromosome number 1, so it will become 4C, right? Now remember this thing, and then we will go in the detail of all of these things when we will do the spermatogenesis and oogenesis. Before that, the last thing about this topic is, in the gametes, suppose that I'm talking about sperm, so it is only the paternal set of chromosomes, so it is only for the chromosome number 1, only one such thing is present, and this is a single chromatid, single chromosome that is present in that. So here I have it is called 1N because they are haploid. There is only the gametes are haploid, very first thing, because there are only 23 chromosomes. They are not 23 pairs, just 23 chromosomes. And secondly, all the chromosomes which are present in a gamete, they are monochromatid, so we call them 1N 1C. Clear? Secondly, the ovum will have same configuration as well, and when the zygote will be formed, so 1N 1C plus 1N 1C, so they will look something like this. Like initially it was the paternal chromatid, the maternal chromatid. When they form a zygote, now for each chromosome we have a pair, that means 2N diploid, and for each chromosome we have 2 chromatids, like one chromatid and another chromatid, so it also become 2C. And when they will replicate, like for the mitosis which will happen throughout the course of development of an embryo, so they will become 2N 4C. But we will see that, don't confuse yourself right now, just take this concept which I have talked about right now, and then we will talk about further from here onward. So here you can see egg becomes zygote, then 2 cell stage, 4 cell stage, then there is morula, and ultimately we have blastocyst, right? So here is another thing, that 2 cell stage, then 4 cell, then 8 cell, morula, early blastula, and then the late blastula with the blastocele. Okay, blastula, the blastula is the thing which has the blastocele. Morula becomes blastula when the blastocele develops, and there is differentiation of this or the segregation of cell in the late blastula. Okay, and then we have zona pellucida, that is the outer covering of the I guess it is outer covering of the zygote or the ovum. I guess zona pellucida is the outer covering of the ovum. So we will continue this. Now let's talk about DNA synthesis. In a zygote, the DNA is 2N, which means we have 2 copies of for each chromosome, and 2C, which means we have total 2 chromatids for a particular chromosome. That leads to, after that there will be DNA synthesis, and duplication of the chromatids will take place in the cell will become 2N 4C. So now it is the same concept. So I will try to highlight it over here. Suppose I'm talking about chromosome number 1 in a zygote, or in a let's talk about zygote. So when, suppose this is zygote, which is formed by the fusion of sperm and the ovum. Now this zygote is containing how many copies of chromosome number 1? That is 2N. And how many chromatids are there in total for the chromosome number 1? They are 2, so this is 2C. So just make this concept clear first. Now whenever the zygote, because zygote is formed, and now the further division in the zygote is going to take place by the virtue of mitosis. So before every mitosis, there will be DNA duplication. So how will the DNA duplication take place? The chromatids which are like this chromatid, that will become duplicated, and this chromatid will also become duplicated. So even this is a chromosome unduplicated, and this is a duplicated chromosome. Both of these things are representative of a chromosome. So now this is the picture just before mitosis, or just before division, when the DNA has when DNA has replicated. So now this particular thing, it is still, there are 2 chromosomes for chromosome number 1. There are 2 copies of chromosome number 1, one from the maternal side and one from the paternal side. So number is 2. Okay, but the chromatids, now we count them, 1 2 3 4, it will become 4C. So 2N 4C is the picture we get before the cellular division. Zygote, and then it divides to form 2 cells, and then again the 2 cells will have 2N 2C. So the baseline, you may say that equilibrium configuration, or the baseline configuration in all of the cells of the body, it is 2N 2C. Exception is in the gametes, where it is 1N 1C. In the case of a cell that is just going to divide, it will become 2N 4C. So these are the small things that you must remember. Then fetal development, 2 cell stage, first 1 to 2 days after fertilization. So in first 1 to 2 days after fertilization, the 2 cell stage is arrived, like the zygote, that will be broken down into 2 cells, like this one cell, add another cell. So this 2 cell stage comes 1 to 2 days after fertilization. Morula, there is a ball of cell without cavity. So the difference between the morula and the blastula is the presence of blastocele of the cavity. So it is a ball of cells without cavity, forms after multiple divisions, and forms at the 4th day of fertilization. Then blastulation, what is that? Formation of a fluid filled cavity within the morula. So suppose that this used to look like something like this, that these are the cells and cells and cells, a lot of cells over there. So it was a tuft of cells, but there was a fluid that was in, some fluid entered this, and then there was separation of the cells, and separation happened in this way, that some of the cells came to the pole, and the rest of the cell made a rim out of that, and in the center we have the cavity. So this cavity is called as blastocele, or the blastocyst cavity. The cells which are surrounding, they are called as trophoblasts, and the cells which are forming this pole, it is called as the inner cell mass or the embryoblast. Formation of the fluid filled cavity within the morula. The cavity is called as blastocele. It forms in the 5th day after fertilization. So on the 2nd day we have like 2 cell stage, on the 4th day we have morula, and on the 5th day we have blastula. Outer cell are the trophoblast, polarized, one side different from the other. Polarized. Okay, so what does polarization mean? Suppose I'm not talking about these trophoblast cells, but in other cells, what do we see is, so elsewhere in the body Suppose that I'm talking about a nephron cell, suppose. So it has like a luminal side and a basolateral side. So it has polarization, like this pole is the luminal side and this pole is the basolateral side. And on the for the nephron, it has sodium potassium ATPase on the basolateral side and some particular channels on the luminal side, depending on the site of the tubule, like if it is a PCT or a PCT cell or the loop of Henle and stuff like that. So forget about that. The thing that I want to mention is that the trophoblast cells are polarized. One side is different from the other. Watery fluid of the blastocyst is secreted by the trophoblast cell. So this fluid is being secreted by the trophoblast cell, the watery fluid. Okay, differentiate into cytotrophoblast and syncytiotrophoblast. The syncytiotrophoblast invades the endometrial connective tissue 6 to 7 days after fertilization and starts secreting the beta HCG, which signals to the corpus luteum in the ovary to continue producing progesterone. So the syncytiotrophoblast invades the endometrial connective tissue 6 to 7 days after fertilization and starts secreting beta HCG, which signals to the corpus luteum in the ovary to continue producing progesterone. So this is very important, that the cytotrophoblast, suppose this is the cytotrophoblast, there will be a picture later on. There is like this one, there's a good picture. So the cyto means intact compact cells. So the cells are cytotrophoblast, while the syncytium, that is the syncytiotrophoblast. So the compact cells, like as you can see in this picture, if the cells are having the intact structure, that is called as cytotrophoblast, and the syncytium of the cells, like the cells lose their integrity and a tuft of nucleus is seen, that is called as a syncytiotrophoblast. Suppose over there, so syncytiotrophoblast is the outer structure. So these cells, some of the cells will grow like this, that the cytoplasmic membranes or the cytoplasmic membrane, they will fuse together and a continuous or a syncytium of the cytoplasm will be formed with nucleus in between, like these are the nuclei. So these nuclear structures are present in between. So this particular thing is called as a syncytiotrophoblast, that is a composite structure. No differentiation of between the cell is present in the syncytiotrophoblast. So such a structure is called as syncytiotrophoblast. And what does it do? Syncytiotrophoblast invades the endometrial connective tissue 6 to 7 days after fertilization and starts secreting the beta HCG, which signals to the corpus luteum and the ovary to continue producing progesterone. So here it is, that the corpus luteum, we will see that when we will do the the growth, the follicular growth and the ovarian cycle and stuff like that. So here you must remember that the syncytiotrophoblast is going to produce the beta HCG, and beta HCG will give the signals to the corpus luteum to continue producing the progesterone, and this beta HCG is also the foundation of the pregnancy test in the urine. Then inner cells, the embryoblast or the polar or apolar, I guess what is apolar, I don't know, but this particular cell, this is the embryo blast. It gives rise to all tissues of the body and comprised of the embryonic stem cells. So this particular thing, this particular pole has embryonic stem cells, and it is where all of the cells of the body will be formed. Like this is the thing that will be divided into the germ layers, the ectoderm, the mesoderm and the endoderm, and all of the morphogenesis will be taking place through this pole. Implantation. The blastocyst implants in the uterus about 6 to 10 days. There was a mnemonic like the blastocyst sticks at the 6th day. So stick and 6th. So like from 6 to between 6 to 10 day, the blastocyst implants in the uterus. Beta HCG secretion begins. Gastrulation. Blastula is a 3-layered structure, and a blastula converts into a 3-layered structure that is called as gastrula. 3 germ layers are formed from the 3-layered structure that is called as ectoderm, mesoderm and endoderm. Steps of gastrulation. Inner cell mass, bilaminar disc. Two cell separated by the basement membrane, epiblast and hypoblast at 2nd week. So it looks something like that. Let me draw the Dr. Najib lectures over here, if I can. So this is the upper layer, and here we have the lower layer, and this is the 3-dimensional picture, something like that. And we have the blastocele or the blastocyst below that, and these 2 layers of the cells are called as epiblast and hypoblast. So this is the initial cell line, or the initial form, or the initial morphology of the gastrula, right? Formation of the primitive streak. Formation of the formed by invagination of the epiblast cells creates a visible line or streak in the blastocyst. Okay, the presence indicates start of gastrulation. So this primitive streak, like that, the one forming over here, That is the foundation or the hallmark that the gastrulation is started. Epiblast, that epiblast converts into three germ layers, that is ectoderm, endoderm and mesoderm. That is a very important point. I don't know if they ask this about in the step 1 exam or not, but you must remember that the hypoblast is completely removed. There is no remnant of the hypoblast in an adult embryo. The only thing that is giving rise to all the three layers is the epiblast. So all of the three layers, like the ectoderm, and then will form the mesoderm, and below that the endoderm will be formed. So these three layers, ectoderm, mesoderm, endoderm, these are formed, all of them, from the epiblast, and hypoblast gets involuted. So here you can see the two cell stage, and that forms the morula, then that leads to the blastula, inner cell mass, embryoblast, blastocyst, cavity, and outer cell membrane, outer cell mass, that is trophoblast. Here we have the amniotic cavity, then epiblast and hypoblast are differentiated. So this is amniotic cavity, and this is exocoelomic cavity, the primitive yolk sac. So on one end of the hypoblast, embryoblast, we have the amniotic cavity, and on the other pole we have the primitive yolk sac, and then the exocoelomic, that is something Heiser's membrane. Okay, then we have epiblast, and then the primitive streak is over there. So primitive streak, the formation of the primitive streak is the indicative of the fact that the gastrulation has started. And then here we have the hypoblast. Then we have it here that just give me a second. Yep. So the blastocyst implantation. So how does that take place? Blastocyst, blastocele and blastocyst cavity, embryoblast and the inner cell mass. The trophoblast become trophoectoderm. So the implantation starts on from 6th to 10th day. Okay. So where's that? Fertilization, and then we have morula. So fetal development, the two cell stages from 1 to 2 days, morula is from 4th day, and blastula is at the 5th day, and then implantation begins 6 to 10 day. Okay, so implantation 6 to 7 days after fertilization, what happens is that the syncytial trophoblast starts impinging on the endoderm, and it starts digesting that, and that digestion, syncytial trophoblast secretes the beta hCG, and this is the decidualized endometrium. Then we have cytotrophoblast that secrete the hCG. Okay, then here we have this is being implanted, and during this time there may be slight bleeding, and that slight bleeding can be confused with the menstrual bleeding, but this is the bleeding of implantation. There is some more technical name to that, but I am calling it the bleeding of implantation, stuff like that. So cytotrophoblast secrete the hCG. HCGH, I don't know what that means, but hCG that is produced by the what? Cytotrophoblast. Okay, no. There are two forms of hCG. The beta hCG is formed by the syncytial trophoblast, but hCGH is formed by the cytotrophoblast. Okay. Then we have the amniotic cavity that forms in the embryonic pole. Then these are the lacunae. Okay. Then the developing villi will also be seen. As you can see, these are the villi, and the maternal blood will pool into the lacunae. Then we have embryonic disc over there. Okay. Then the yolk sac forms, and then we have hyperglycosylated hCG will be secreted in this phase. So cytotrophoblast is forming hCGH, syncytial trophoblast is secreting the beta hCG, and the fully implanted uterus will be forming hyperglycosylated hCG. Summary of the early fetal development, that is from FA2 2018, within week 1, week 2, within 3, 3 to 8, 4, 6, 8, 10. Okay. So let's see it one by one. Okay, so within first week, hCG secretion begins around the time of implantation of the blastocyst. Blastocyst sticks at the day 6. Then within week 2, bilaminar disc, epiblast and hypoblast. 2 weeks is equal to 2 layers. Then within week 3, okay, so within week 1, the hCG secretion begins, and blastocyst sticks at day 6. Remember that. Then within week 2, the bilaminar disc forms, that is the epiblast and the hypoblast one. So 2 weeks is equal to 2 layers. Within week 3, gastrulation forms trilaminar embryonic disc. Cells from the epiblast invaginate. They will form primitive streak. Endoderm, mesoderm, and ectoderm. Notochord arises from the midline mesoderm. Overlying the ectoderm becomes neutral plate. So 3 weeks is equal to 3 layers. So let me explain this concept. That suppose this was epiblast, hypoblast gets involuted. So I am not drawing that. This is epiblast over here like this, and this is a structure, something like that. So there forms a primitive streak. Now the primitive streak is formed. It is a compact structure. So throughout the course of embryology, you will see that firstly there is a rigid structure, and then the cells in the middle they die out due to apoptosis, and that rigid structure becomes a structure having a lumen. So same happens in the case of a primitive streak, that is cells which are in the middle of the primitive streak, they die out, and we get a uh, primitive groove. So primitive streak becomes primitive groove. Now this is epiplast, right? The epiblast cells becomes ectoderm, and the cells which are present like over the surface, they start moving in this direction. They start dipping through the primitive groove downwards, and these cells which are dipping from this primitive groove downward, they will form ectoderm, they will form endoderm, and in the middle they will also form mesenchyme. So all these three layers are coming from the epiblast. Remember this one. So the primitive streak, ectoderm, endoderm, mesoderm, and ectoderm are coming because of the epiblast invagination. Notochord arises from the midline mesoderm. Overlying ectoderm becomes neural plate. That is beautiful concept. That suppose that after the formation of, like, this is ectoderm overlying, there is the ectoderm like this, and here we have endoderm below that, okay, with a amniotic cavity, and this is the endoderm. In the middle we have mesoderm. Like this is the mesoderm, and this is the mesoderm. This particular dot is a notochord. So that notochord is formed from the mesoderm. Now this notochord, this is from the mesoderm, right? The notochord arises from the midline mesoderm. Now this notochord will give the signal upwards and sonic hedgehog signaling, to be specific. Those sonic hedgehog signaling will be received by the ectoderm that is lying over the notochord. So notochord is basically a tube. So definitely this longitudinal structure has a specific chunk of the ectoderm that is overlying this through its length. So that ectoderm, that part of the ectoderm that is overlying this particular structure throughout the course of its length, that becomes the neuroectoderm. So this particular area of the ectoderm becomes the neural plate, and then neural plate will become, neural groove, and then neural groove will become neural fold and stuff like that. So that the process of neurulation starts like that, and that begins within the 3rd week. Notochord arises from the midline mesoderm. Overlying ectoderm becomes neural plate. Till the formation of neural plate, we have till the 3 weeks. Then week 3 to 8, it is the embryonic period. Neural tube formed by the neuroectoderm and closed by week 4, and organogenesis takes place. So the neural tube is formed, and neural tube is basically it has two ends. Like, suppose it is the neural tube, so it is an anterior neuropore and the posterior neuropore. So these two pores are supposed to be closed. If the anterior neuropore does not close, it will lead to anencephaly, and that particular condition is not it's not fit for life. The patients are not viable. The fetuses which are born, they are not viable. And in the case of the posterior neuropore, if that does not close, the person can develop spina bifida, either occulta or other conditions like that. So these are the things that can be formed. So this is the about 3 to 8 week embryonic period. Okay. So neural tube formed by the neuroectoderm and closes by week 4 and organogenesis. It is extremely susceptible to the teratogenesis. So this is the phase, and they ask you a lot about this one, that in which phase the teratogens have their maximum impact. So that is during the week 3 to 8, and it is extremely susceptible to the teratogenic effect. Then in the week 4, 6, 8, they have also given it a separate thing. So heart begins to beat in the week 4. Upper and lower extremity limb buds begin to form. 4 weeks, 4 limb and 4 heart chambers. In the week 6, fetal cardiac activity is visible by the transvaginal ultrasound. In the week 8, fetal movement start, gait at week 8. And the week 10, the genitalia have male or female characteristic. Prior to week 10, genitalia look similar for male and female. SRY gene of the chromosome Y cause the penis development, and lack of SRY gene cause the clitoris development. Ultrasound and identification of the gender usually week 15 to 20, and tenitalia. So within the week 1, blastocyst sticks at the day 6. The very important or the hallmark finding is that within 2 weeks we have 2 layers, that is epiblast, hypoblast. Within 3 weeks we have 3 layers, like 3 germ layers formed. Along with that, the neural plate is formed. Week 3 to 8, the neural tube is formed, neuropores are closed, and organogenesis starts taking place. In the week 4 week, the limb buds, like 4 limbs and 4 heart chambers. In the week 6, fetal cardiac activity is visible by the transvaginal ultrasound. In the week 8, Fetal movement starts gait at week 8, and week 10 we have genitalia, and you can say them tenitalia. So the presence of SRY will cause the formation of penis, and the absence of SRY will lead to the formation of clitoris, and ultrasound identification of the gender usually week 15 to 20 can be done. Then embryologic derivatives. So these are very important, and I will recommend you the video of Dr. Nandan Joshi. He has also done like remarkable work about in this thing, like the read with me session for the first year 2022, and he gives like very good mnemonics for this one. So surface ectoderm, epidermis, adenohypophysis from the Rathke's pouch, lens of the eye, epithelial lining of the oral cavity, sensory organs of the ear, olfactory epithelium, inner canal, below the pectinate line, parotid, sweat, and the mammary glands, the craniopharyngeum, benign Rathke pouch tumor with cholesterol crystals and calcifications. So cholesterol crystals and calcification are present in the craniopharyngioma. I don't remember those mnemonics right now. So the mnemonic for this one is clean the surface with soap. Let me write it over here if I can. Okay, my laptop is a bit slow. So but the mnemonic is clean the surface, like it is CLEN, clean the surface with SOAP. Okay, this is about it. Let me make it small. It's too small now. I guess it's good enough. And this is the mnemonic for this one. Clean means C is for the canal, that is anal canal. Then we have L is for the lens. Then CLE is for the it is for E is for the epidermis. Then CLE, A is for the, where is A? A is for the adenohypophysis. And then CLE, N is for the nipples, or you may call them mammary glands, NIPPLE. That is basically gland. I'm typing and this is not writing anything at all. I guess it is held somewhere. So yep, adenohypophysis, and here we have the mammary glands. So this is the mnemonic clean, and now let's move to the, so this is clean, and what comes with the soap? Soap is it is sensory organ, no, it is sweat glands. With the O we have olfactory epithelium, SO. Then for the A, the sensory organ of ear, we can write it auditory apparatus, SOA, and for the P we have parotid. So remember it with that, and thank Dr. Nandan Joshi because he has made some great mnemonics. With the neural tubes, the derivative of the neural tube are the brain, neurohypophysis, CNS neurons, oligodendrocytes. Okay, so this is the soap. Neural tube, the brain, neurohypophysis, CNS neurons, oligodendrocytes, astrocytes, ependymal cells, pineal glands, retina, and spinal cord. So neuroectoderm, think CNS. So pretty much everything that is present within the CNS is coming from the neural tube, the brain, the spinal cord, and everything in between. So the brain and spinal cord. Okay, then brain contains neurohypophysis, the posterior pituitary, and then the CNS neurons, which are oligodendrocytes, astrocyte, ependymal cells, and the pineal gland. So the only odd creature among them is the retina. So the retina is basically derived from the neural tube. Then about the neural crest. So the neural crest is it is mortal pass, or I guess in the first year 2022, what was the mnemonic for that? Give me a second. I wrote them down in the annotated now. No, they are here. They are here. So it is Neosporin, the tube of Neosporin. So if you want it, we can make it over here. So for the neural tube, we have the tube of Neosporin. Let me zoom it. So the tube of Neosporin, it means neural tube, epidermal cells, astrocytes, spinal cord, pineal gland, oligodendrocytes, retina, and neurohypophytes. But it is very clearly evident that the CNS is coming from the neural tube. So CNS contains the spinal cord and the brain mainly, and then with the spinal cord and the brain, we have the structures which are present in the brain, like the posterior pituitary and the CNS neurons and blah 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 blah, everything. So you remember this table. This is important neural crystal. What is the mnemonic for that? It is Elmopasses. They have written it like that, and Dr. Nandan had a better mnemonic. It was I don't remember it right now. It was like mortar and something. So Elmopasses, what does that mean? So Elmopasses, so basically melanocytes, you must remember this one because it is very important. Then myenteric or Auribac plexus, odontoblast, endocardial cushions, laryngeal cartilage, parafollicular C cells of the thyroid. Then we have PNS, dorsal root ganglia, cranial nerves, autonomic ganglia, adrenal medulla, and all the ganglia, spiral membrane, aorticopulmonary septum, Schwann cells, spine, and arachnoid, and the bone scale. So let's write the Elmopasses. Neural crest, think PNS and no neural structures nearby. So whole of the peripheral nervous system is coming from the neural crest cells, while the central nervous system is coming from the neural tube. So let's write it over here. It is Elmopasses, right? So Elmopasses, if I'm not wrong. So Elmopasses, the ELMOPASSES. Okay. So Elmopasses, this is the mnemonic, and now let's start writing it down. E is for the endocardial cushions, endocardial, CUSHIUNS, endocardial cushions, then L is for the, let me make the spelling correct, endocardial cushions. Then the L is for the laryngeal cartilage, okay. M is for the melanocytes, O is for the where is O? Odontoblast, odontoblasts. So Elmo, and then for the passes we have they have made two P's, but okay, we will also write with two P's. So P is for the parafollicular C cells, parafollicular cells. Then the other P is for the, where is the other P? There's PNS, like dorsal root ganglia, cranial nerves, and the autonomic ganglia. So PNS. Then A is for the adrenal medulla. Actually, you may not be seeing anything written over there. Okay, now you can see it because my laptop is a bit slow. So adrenal medulla, PA. Then S is for the spiral membrane, okay, spiral membrane, aorticopulmonary septum. So in the aorticopulmonary septum. So remember from the CVS unit, it is basically a tie in the CVS unit. So it goes something like that. That suppose that this is a developing heart. I always draw heart like that, and from it there originates a single artery. A single trunk is originating, and that single trunk is the conus arteriosus. It's not conus arteriosus. It is the common. It is truncus arteriosus. The common trunk that is coming out of it is a truncus arteriosus, and it has a spiral membrane inside it, which causes the division into the division of the truncus arteriosus into the pulmonary artery and the aorta. So if the spiral membrane is defective due to some reason, Resultantly, the person may develop the newborn may develop what? The newborn can develop that thing that we call the transposition of weird arteries. Yes, transposition of weird arteries can transposition of weird arteries, or patent truncus artery. So these things can be formed in the patients with a defect with the spiral membrane. So this is the spiral membrane, and the last test is for the for this one. The last test was for the Schwann cells. Schwann cells. So remember these all things, and that is Elmo passes, or the mortal pass, or something. The way you want to remember it, you can. Then the mesoderm derivatives and the endoderm derivatives are there. I know that we are like ahead of our time. Not ahead of our time. We have crossed the upper limit, 50 minutes. We are at 54 already, but I want to complete this, so let's do it. So the mesoderm, the mnemonic is vectral, or you can also use it clear TV, as Dr. Nandan says. So muscle bone connective tissue dermis, serous lining of the body cavity filled with the peritoneum and the pericardium and the pleura, spleen derived from the foregut mesentery, cardiovascular structure for the lymphatics or bloods, wall of the gut tube, upper vagina is coming from the mesoderm, then kidneys, adrenal cortex, testes and ovaries. Notochord induces ectoderm to form the neuroectoderm, neural plate. Its only postnatal derivative is the nucleus pulposus of the intervertebral disc. Then here we have the middle meat layer, mesoderm. It's not the mnemonic for that, but if there is mesodermal defect, that can cause a vectoral association. So vectoral association, there can be vertebral defects, anal atresia, cardiac defects, tracheoesophageal fistula, renal defects, and limb defects of bone muscle. So we will study that later about the associations. But the thing is that all of these things, they happen simultaneously due to some unknown reasons. They are found to be happening together. There is no pathological or pathogenetic reason behind them to happening together, but they are seen to be happening together in the fetuses. Then we have the mesoderm over here, and in the mesoderm we have like the derivatives of the mesoderm are the middle layer of the meat. So muscles and the bones and connective tissue and dermis, along with the serous lining of the body, peritoneum, pericardium, pleura. Spleen is coming here. That is very important, that although it is coming from the mesoderm, it gets its blood supply from the foregut celiac artery. Okay, then cardiovascular structures. Yep. The lymphatic blood, wall of the gut tube, upper vagina, kidneys, adrenal cortex, testes and ovaries, and even the notochord itself. Endoderm of the antral layer, the gut tube epithelium, including the anal canal above the pectinate line, because we saw that the anal canal below the pectinate line was coming from the surface ectoderm. Here we can see that the anal canal above the pectinate line is coming from the endoderm. Most of the urethra and the lower vagina derived from the urogenital sinus. So the lower vagina is coming from the endoderm, while the upper vagina was coming from the mesoderm. Okay, remember this one, and here we have this one. So derived from the urogenital sinus, the luminal epithelial derivatives, for example, the lungs, liver, gallbladder, pancreas, eustachian tube, thymus, parathyroid, and thyroid follicle cells. These all things, like luminal epithelial derivatives of the lungs and the liver and the gallbladder, pancreas, eustachian tube, thymus, parathyroid, and thyroid follicle cells, all of these things are coming from the endoderm. Then we have the organ with a special embryologic origin, so we will talk about them later on. So until then, Allah Hafiz, fi amanillah.